We were on. Okay, we were talking about this. This was the last thing we ended on, right? Yes. But I want to go back and we're going to talk about this real quick. But yes. Full screen. Okay. And I need. Okay. How are we doing? Oh, we still got a few minutes. So go and make sure that you did your seating chart. So go and make sure that you did your seating chart, not only for today, but also Friday. Fridays looked kind of bare to me. I don't know. Just make sure your Friday one is correct, but this is today's. So y'all notice at the bottom, there are tabs and there's a tab for every day, right? But I'll always put today's first, so it's easy for y'all. Okay. The wrong thing here, this one. Okay. We still got 2 minutes. Just. So everybody had a good weekend?
Absolutely.
Got some sleep, some rest, some relaxation. I started watching The Crown. I think I'm going to be addicted.
It's good.
Yeah, it's on Netflix.
After like season 2, the cast changes, so they all get older,
Yeah.
and it's not as good. But Princess Diana is.
Okay. All right. Something to look forward to. I'm like, this is going to be bad. But I needed a little something to watch, a little something to do. That wasn't schoolwork, right? All right. Okay, so let's see. Today's joke is how do monkeys get down the stairs? They slide down the banana stairs. I had to explain to Charles what a banister was. But that's okay. Like I feel like even when he doesn't get the jokes, it's a language opportunity to teach, to expand his vocabulary. So, you know. Squid, what is Squid Game? I don't know what that is. All right. So we were talking last time about protein purification, right? So what's the name of the game? What did I say the name of the game of protein purification is? You want to increase the purity of your sample so that you have fewer and fewer different things inside of it. And what else? And increase the concentration. So you're going to increase purity and concentration. And so the way that we signify that is we look at what's called specific activity. So if we step back one slide, right? Increasing purity and concentration is a way to say enrichment or specific activity. It's the same thing. So you're looking at two things for specific activity: the total amount or activity of the target. How do we determine this? Oops. How do we determine that? Oh, come on. So how do we determine this? We do the what? Well, we do an activity assay. Activity assay. What activity assay did we list before? If you go back. Go back here. Right? This is the luciferase assay, but it's really luciferase activity assay, right? what you're doing in this assay is determining in your samples, does your sample have enzymatic activity of this one particular enzyme, right? Y'all with me so far? Okay, so to determine specific activity, we need to know the general activity, and then we're going to divide by the total amount of protein in the fraction. So let's say at the beginning, we have our whole cell lysate or our homogenate, homogenate, right? Our whole cell lysate. All you've done is broken open the cells. Would we have activity? You have activity. You broke open the cells. Is your protein present? Yes. So if you tested it in an assay, would it have activity? Yes. So I'm going to put a yes here. But tell me about this total protein. Protein, protein. Do we have a lot or a little? Of total protein? If I just break open the cell, it's all there. I'm going to have a ton of protein in the sample, right? So I have lots of different proteins, right? So when I look at that, those two things, I'm going to take the activity and I'm going to divide it by the total protein. So I'm going to skip ahead real quick. I'm going to skip ahead a lot, but here, where we look at the total protein. So here's my preparation of crude cell extract. I look at the total protein. Do I have a lot of protein? Absolutely, 2,000 milligrams. I can look at total protein by just looking at absorbance of my aromatic residues. Right, I could do that, or I could do a BSA standard curve like you're doing in class, right? All of those are different ways to assay just total protein. So I look at total protein and then I look at activity. Well, if all my proteins are there, I'm going to have quite a bit of activity. But if I take my activity and I divide it by my protein, what do I get? A specific activity of what? 10. That's not very high. If I do one step, if I do subcellular fractionation using differential centrifugation, right? And I collect just my one sample that I want. So in my case, I wanted that cytosolic fraction, right? Y'all remember that? What happened to my total protein once I pulled out the mitochondria, pulled out the nucleus, pulled out all the other parts that I didn't want? What happens to my total protein? To go up or down? Goes down. What happened to my total activity? Pretty much it stayed pretty close to the same. I lost a little bit. You're right. I lost a little bit of activity. So some of my protein might have been trapped somewhere and got pulled out. But my activity is pretty close to the same, and I've cut down my total protein by how much? By about a fourth, right? So if I calculate my specific activity by taking 18,000 divided by 500, what happened to my specific activity? It increased. The name of the game, this is the name of the game, to increase your specific activity. Because that takes into account the two things that I want. I want to increase the concentration of my protein of interest, and I want to increase the purity. So I have fewer other proteins there. That's the definition of specific activity, just using more common terms, right? So what you can do is you're going to look and you're going to see how every step in our purification scheme is going to increase the specific activity of our protein of interest. And that's a good purification scheme, what we're looking at right here. And we should see a fold increase in purity at each step, a fold increase, right? That's a lot. So how do you calculate your fold increase? And if you look, do you see how these have little subscripts, superscripts, excuse me? This is B, it tells you. Multifold increase is calculated by dividing the specific activity at a given step by the specific activity from the previous step. So 36 divided by 10 is 3.6. You will have to do these calculations in lab. Okay. All right, so let me go back. Because we're only in the very beginning of purification. All we're doing right now is preparing different fractions, right? So we spun the first time, we got a nuclear fraction, but we don't want that, so we threw that away. We kept the supernatant. We spun it again, faster and longer. We pelleted the mitochondria, but we don't want that, so we got rid of the pellet. We kept the supernatant. So we spun that a little bit faster for a little bit longer. We got a membrane fraction. Do we want that? No, get rid of that. We want to keep the cytosolic fraction. So when I look at this, right, my initial, I had 20,000 activity units, but I had 2,000 milligrams of protein. Good Lord. After collecting just this cytosolic fraction, I now have pretty close to the same activity, a little bit lower, but pretty close to the same activity. And I have 500 milligrams of protein, so much less protein total, but more of that protein is my protein of interest. So that's why I was able to concentrate what I wanted, right? So we can calculate specific activity for both and then divide 36 by 10 and get the 3.6-fold purification, okay? You have to be able to do those calculations. It's not hard though. It's just division, right? It's not hard. You just have to know what's what.
Wait, what are you dividing 36 by?
So in order to get the fold purification, you take the specific activity from one divided by the specific activity from the previous step. So 36 divided by 10, you get 3.6. And the explanation for the calculations are on that slide with the table. Okay. All right. So you have centrifuged your sample, right? You have the fraction that contains your protein of interest. In our case was the cytosolic fraction. But still within that cytosolic fraction, how many different proteins are in the cytosol of a cell? Still a bunch, right? Still a bunch. So we need to start taking into account some of the other characteristics that are unique to other proteins. One of those things is solubility, right? We talked about how aggregated proteins will precipitate out and that kind of thing. So this is a similar thing, but what we do is we on purpose make our proteins precipitate. So we add increasing amounts of a saturated salt solution to our protein sample. So our saturated salt is ammonium sulfate. Ammonium sulfate is what we add to our samples to salt them out. So every protein has a unique solubility. So maybe protein X will precipitate at 20% salt solution, right? But I'm looking for protein Y, and protein Y will precipitate at a 40% salt solution, right? I can, let's see, we'll do 3, X, Y, and Z at a 60%, let's just say, okay? If I want to purify for protein Y, this is my protein of interest. What I'm going to do is kind of stepwise. I actually want to get rid of things that have a lower solubility and get rid of things that have a higher solubility. So the way that I do that is I would add salt to maybe 35%, right? What would happen when I do that? What would precipitate out? X would precipitate out. I'd be left with Y and Z, right? Okay, now what I can do is I can spin and I can get rid of the precipitated stuff and I'm left with the stuff that's still soluble, right? I add more salt, add more salt, right? As I keep adding salt, things are going to precipitate. So what do I want to do? I want to add more salt to what? 40%, right? So what happens at 40%? What's going to precipitate? Y, but not Z. So now I spin again, and you're gonna have to spin, right? Now I spin again, and what do I keep? Nope, pellet. On the top, you're gonna spin and you're gonna keep the soup. On the bottom, you're gonna spin and keep the pellet. Because what's in the pellet is what precipitated. Well, what precipitated at 40%? Protein Y, which is what I want, right? Did protein Z precipitate? No, it's still in the supernatant. And I'm going to dump that down the drain, right? So it's a way to select out the things I want. But there's a problem with this. I have precipitated out my protein of interest. Is it functional? No, it's precipitated, right? It's in a ton of salt. You do not want it to be in salt for very long. You need to find a way to now separate your protein from the salt. So you do that with dialysis. Okay. So in dialysis, right, what we want to do is we want to remove the salt, the ammonium sulfate, and we want to keep our protein. So what we do is we put our sample that has both the salt and the protein in what's called dialysis tubing. Dialysis tubing, you see how it kind of looks like a candy, right? So it's a tube, and you pipette your liquid into the tube, and then you clamp either end. And so this tube is a porous, P-O-R-U-S, porous tube. Why is that important? That it has a pore. Right? The pore size inside of the tubing is going to allow certain things to exit, and it's going to keep certain things in. How big are salts? Salts are tiny, right? But our protein of interest is huge comparatively, right? So this is our protein, and these are going to be salts. What happens if I put this into a solution where the water here, the water has less salt than the sample? What process happens? Diffusion happens, right? Where's the salt going to go? It's going to go to the area of low concentration. So all these salt molecules are going to diffuse out of the tubing. Is your protein going to go anywhere? No, your protein's stuck inside this dialysis tubing. So this requires a large volume of buffer. So this buffer right here, you need a large volume. Otherwise, it doesn't work, right? Because you need to be able to pull out all the salt. What happens once you pull out all the salt? The protein is now going to renature and fold properly, right? So you have to be very careful that the buffer that you put it in is a large volume and it has the correct other characteristics like pH, like ionic strength. Some proteins require a little bit of an ion, right? Maybe a magnesium or a calcium ion. Some of them require something. So you have to be very careful that the buffer that you dialyze in is the perfect environment for your protein of interest, because that's what will end up in the dialysis tubing. Make sense? So what's the basic concept that dialysis is based on? Diffusion, simple diffusion, but it works. Okay. All right. Trying to find mine. There we go. All right. So what we're doing each step, we did differential centrifugation, right? Let's do our little pathway first. So protein purification. The first thing you do is open the cells, right? In all the different ways that we talked about. sonication, what else? Shearing, detergents, right? Okay, what happens after you open the cells? What are you going to do? Open cells, then what?
Separate the protein of interest.
So the whole point of the rest of the scheme is to separate out our protein of interest, but what we want to do is a stepwise process to make that happen. So I want to get out some big things. How do I do that? Differential centrifugation. CNTRIFGUATION? I don't think so. 3. What happens after differential centrifugation? Look back in your notes real quick. Salting out. So this is solubility differences. Right? So every time we go through a new step, we're going to separate out more of what we don't want and keep what we do want. So what do you think the next step is? Column chromatography. Oh, but wait, before we do that, we got to do what? Dialysis. You got to get the salt out. MATOGRAPHY. Right? So if you skip ahead and you go to the table, can you see everything we've listed, right? We prepared the crude cell extract, then we did centrifugation, then we did the salting out, the ammonium sulfate precipitation, and now we're going to talk about the different ways to do chromatography. So the ways that you can do column chromatography, there are 3 different ones: gel filtration, ion exchange, and affinity. And they're listed in this particular order because they provide a stronger and stronger amount of separation. So they're listed that way in a particular order for a purpose. Okay, so we're going to talk about them hopefully in that order. I did it right. Yeah. Okay. All right, so we said the 3 of them: ion exchange, gel filtration, and affinity. And they're actually in a different order in the slides, but that's okay. Ion exchange, what do you think that's going to be dependent upon? Right, remember how I said every one of these steps is dependent upon a characteristic of your protein of interest? So what characteristic of your protein of interest do you think ion exchange is going to be dependent upon? Charge. Depends on charge. Okay, affinity. When we talk about affinity, a lot of times it's binding affinity. Have you heard that before? So what do you think affinity is dependent upon? Who your binding partners are and how tightly you bind to them, right? So this is binding, we'll just say binding characteristics. So partners and how tight is the binding. Okay, now gel filtration. The term doesn't give away the answer. Gel filtration is all about size, right? How big your protein is. And we're going to talk about why that is. Okay, good. All right, so no matter what type of chromatography you're talking about, you're going to use some physical or chemical property of that protein, of how that protein interacts with whatever is inside the column. And we call what's inside the column the matrix. Okay, so you have your protein sample and you have your matrix. And whatever your protein sample is will have properties, and whatever your matrix is will have properties. And how they interact is going to separate out the different things inside your sample. Okay, so if we look at a general chromatography technique. I'm not telling you which one it is, but this is, doesn't matter what it is, whether it's affinity or gel or whatever it is, this is how you're going to set it up. You have a column, and inside that column, you have some kind of a matrix. And the type of matrix will depends on the column type. So it could be beads of different sizes. It could have charged items, CHAR, charged species. It could have many different things. It could have proteins attached. Could have all kinds of things, but it's all going to be dependent upon which kind of chromatography you're doing. But we know in the beginning, we're starting out with a tube, right? Here's our tube. And this is our protein mixture. Because we just did the salting out, we just did dialysis, we're going to have a couple of different proteins inside of our sample, right? We're not completely pure by the time we've done our salting out step. We still have a mix. So we're going to take that mix and we're going to pour that mix onto the column. Now the column has buffer in it already, the same buffer that your protein mix is in. And then what you do is you take more buffer and you put it on top. It's called the running buffer. And what that does is you have your protein and it's sandwiched in between buffer. And the more buffer you add, it pushes your sample down. Now, however much the samples interact with the column will determine how quickly they move through the column. So what happens if they don't interact with the column very much at all? They run right through. So that's the black one. The black one came out first. The red one interacted a little bit, so it comes out second. And the blue one comes out last because it interacted the most. And what you do is you collect fractions, maybe 1 milliliter fractions at a time, so that you get a separation, right? This is perfect that you have each one in one tube, but it doesn't always work out that way. But this is optimal, right? Okay, so let's talk about some of the individual ones. Oh yeah, their animation on this is really nice. Y'all are watching the animations, right? They're really helpful, aren't they? I think they really are. Okay, so if we look at a basic scheme of what happens, right? What we want are for things that don't interact with the column to go through very quickly. And it's probably going to be a bunch of different things that are going to come through in the beginning, right? Because it didn't interact with the column. So we have lots of other proteins coming out here. But what we want to see is that later, all the junk proteins have come out and our protein of interest finally comes out at the end, right? So this would be the black protein and the blue protein. And this would be the red protein coming out, right? Wasn't that the color scheme that they did? Oh, no. So they should have done that differently, right? Their protein here was the triangles. We'll put it that way. So this would be the triangles. And this was the circles and squares. And squares, right? Because the circles and squares didn't interact very much, so they came out early. The triangles interacted a lot and they came out a lot later. So if you look here, these are the fractions that came out. Now, we can look at total protein concentration. That should be very high here because our circles and our squares came out here. And you're going to have relatively low concentration here. But of that stuff, almost all of it is your target protein. So what should my activity look like for my early fractions? Really high or really low? It should be really low. It shouldn't have much activity at all. But what about when I get to the point where my target protein comes out of the column? I should have lots of activity, right? And I do. So in my later fractions, 6 looks like 7, 8, 9. These fractions have my triangles in them. And so I want to keep those. but I don't want to keep these early ones because that's got the circles and the squares that I don't care about. Okay. All right, so let's talk about each of the different kinds of chromatography. So the one that gives you the least amount of separation is gel filtration. And what did we say gel filtration was based on? Size. Could you have a lot of different proteins that have similar size? Absolutely. So you're not going to get a huge change in specific activity with gel filtration. You'll get some, but not as much as the other two chromatography types. So you might see it listed as size exclusion chromatography, but it's the same thing as gel filtration, exactly the same thing. So if you look at the matrix, do you see the white? This is the white. That is equal to the matrix. That matrix, they're beads, but they're not solid beads. They look more like sponges, right? They have tons of teeny tiny holes in them. And I want you to think about it like an obstacle course, right? You could put me and my 9-year-old, right? And if we had to go through an obstacle course, who would get there first? My 9-year-old would, right? But what if, because I'm so big, I can just go on the side of the obstacle course. I don't have to go through all the little weaving and do this and do that, right? And he does. I can just straight shot to the finish line. Who's going to get there first? Theoretically, me. I'm gonna have to test that one out. But the little ones, the little proteins have to go through each and every one of these little holes in the matrix. And so it slows them down. These large proteins, they just speed through. They go on the outside of the beads. So they end up eluting faster. So which proteins had more interaction with the matrix? Small proteins or large proteins? Small proteins had more interaction. So small proteins had more interaction with the matrix. So came out later, right? So came out later in a later fraction. So the more interaction, the slower they go through the matrix. And in this case, it's do the little things go through. Now you can buy matrix with different size holes in them so that you can get really specific in which proteins you want to slow down and which ones you want to let go through. Because maybe you're trying to purify a really tiny protein. So then I want really tiny holes in my matrix so everything else will run out. And then my tiny protein will take forever to get to the bottom. Yeah. All right, so have you heard of HPLC before? Yes, no, maybe? So it's the same thing. It's called high performance liquid chromatography. Did we just do liquid chromatography? Was our sample in liquid? Right, it was in a liquid buffer and we poured the buffer on top of the column. It's the same thing, but the difference is that the matrix, the particle size is so small that it's so dense that if you just poured liquid on top of it and walked away, it wouldn't do anything, right? So what you have to do is you have to put pressure and force the liquid through the matrix. And so that's why they call it high performance liquid chromatography. And so you can separate out things that are very, very small from one another with a really small size difference because of this. So you might be able to do that one day. It's really cool. Okay, so our second kind of chromatography is ion exchange chromatography, right? And what do we say this is dependent upon? Charge differences, right? Well, you kind of only have one difference, right? It could be positive or it could be negative, right? So basically your matrix can be one of two things. Your matrix can be positive or your matrix can be negative. So let's say you have a positive matrix. You have the DEAE matrix. What you have is a solid bead, and attached to that solid bead is a positively charged group, right? So what's going to bind to this? If I have a protein that comes in, what kind of a protein is going to bind? A positive or a negative? A negative is going to bind. Okay, so if I have a positively charged resin, it's going to interact with a negatively charged protein. This is my protein, right? What if I have a resin, the CMC resin? This resin has a negative charge on it. So what kind of a protein is it going to bind to? A positive, right? Now, it's bound. It's stuck on there. What do I do? But if I take out the resin, my protein's still attached to the resin. How can I detach my protein from the resin? I need to put something in the column that has a negative charge, right? A stronger negative charge than my protein of interest. Could I use ions that have a different charge? Like if my protein is an overall +1 or +2, can I use an ion that's a stronger charge? Absolutely. A stronger salt concentration would work, right? So you have different ways that you can elute your protein of interest, right? So here's an example, right? And these are really two different columns. I don't know why they put them on the same one, but it's two different columns. So this is what kind of a column? So go back. It's a positively charged anion exchange. So this one's the anion exchange. Ooh. What's this one down here? Cation exchange. When we name it, what do we name it according to? We name it according to charge. Oh no, they didn't, they changed this one. Oh, they changed it. Okay, never mind. They're just showing you one. We'll draw the other one. Okay, it's an anion exchange. So you load your protein sample on here and you have two different things going on. You have positively charged substances, right? And you have negatively charged substances. So what's going to interact with the beads? The opposite, whatever it is. So if my beads are negative, my positively charged is going to bind and it's going to stick to it, right? What happens to my negatively charged particles? They just go through and eventually they elute out, right? Negatively charged proteins elute out and we collect them. But I still have my positively charged proteins associated with the column matrix. So what do? I add an ion. So the ion is of similar charge as the protein, right? And we call this the elution buffer. The elution buffer has some kind of an ion that's similar in charge. So it's a positive, whereas our protein of interest is also a positive. Usually we increase the concentration or we make it something that's like a +2 or something like that, but it'll still work. So what happens is that these ions will compete with binding to that negatively charged bead, and then your positively charged protein of interest will elute out. But what's the problem? Now what do I have at the end? I have my protein of interest. What else do I have in my sample? I have these sodium ions, right? What if my protein's not happy in sodium ions? What should I do? Dialysis. That's right. So sometimes you have to do multiple dialysis steps just to make sure that your protein stays in the correct buffer the whole time. Otherwise, maybe it'll precipitate, maybe it won't function. Okay, so how would you describe this if you were doing the positively charged? Let's do a positively charged column. We have beads, and what's on the beads? These are all positive charges. So what's going to bind? proteins that are negative are actually going to bind. What's going to happen to, whoa, I put a positive sign there. What's going to happen to my negatively charged? I'm sorry, what's going to happen to my positively charged proteins? They're going to elute out early. So these elute out early, right? Now what do? I'm going to add something that's negatively charged. What could I do? Anything that's negatively charged is going to compete with the binding site, right? So then we have our protein of interest will elute out because I have ions. So maybe they're the little dots, right? That will compete for the binding sites. Right? Then I can elute out my protein of interest. It's the same thing, just opposite charges. Okay. All right. Last type of chromatography, affinity chromatography. So affinity chromatography is the most specific because if you think about it inside of the cell, most proteins have certain interaction partners, and those are the only other proteins that they interact with on a regular basis, right? They have best friends in the cell and they only hang out with those particular other proteins. So this you can get really, really high separation. So you want to pick something to attach to your bead that your protein of interest has a really, really high affinity for. And you covalently link that new protein, that binding partner, to the bead. And then you put your protein through the column, and then, right? So you have your bead, and your bead has the binding partner, that's kind of the gray part, right? And you actually covalently link it to the bead so that you can't disrupt it. You pour your mixture over, your target protein, the yellow one, will bind. These other proteins, the other junk that you don't want, won't bind, so they'll elute out, right? So it's really important to pick a very specific interaction. Then what you need to do is you put in free competing protein of interest, right? Your binding partner to the protein of interest. If you have binding partners that are no longer bound to the matrix, you can elute that out. But you have to remember, what do you get at the end? You're going to get your protein of interest that's bound to a ligand, right? So this is the ligand, and this is your protein of interest. So you do end up with a protein that's actually bound to something at the end, and you might not want that. What could you do? Sometimes just changing the salt concentration will disrupt the interaction, and then you can separate them that way, right? But you got to always think that sometimes purifying it creates another problem, right? Then you have to dialyze it out again. I mean, you know, it's a very complicated and very stepwise, precise method that you need to do to purify particular proteins. Okay, so this is what we get at the end. So affinity chromatography is usually the most definitive way that you can separate things, but you don't want to put a sample of a bunch of different proteins on an affinity column because it kind of clogs it up. So we try to do other purification steps ahead of time, so when we get to the end, you're not putting that much on the affinity chromatography column. So when we go to lab, we're going to stop either here or here. We'll see where the semester takes us. I'm not making promises, but we'll go all the way to either the salting out step and dialysis. We might get to gel filtration. We will see. Okay, but it's one of my favorite things to do. So what do you get for a protein fold difference when you go from just precipitating out to picking a column? Do you see how you have a huge increase in fold purification? Column chromatography does that. That's where you're going to see the biggest differences in fold purification. Okay, so you've purified your protein, or so you think. How do you know for sure? theoretically, if you know what protein it is, you know what gene it's from, you can predict its size, right? You know about how big it should be. So you can check it on a gel electrophoresis. So anytime you're going to do gel electrophoresis, anytime, anything that you put on that gel, you're just checking the sample. You're not purifying anymore. Purification is done. This is a check, not a purification step, right? We're done purifying. We're just checking how effective was our purification scheme. So when we do gel electrophoresis, you have what's called polyacrylamide. And polyacrylamide creates a matrix, right? And I like to think of the matrix as spaghetti. You will add polyacrylamide to like DI water, right? And you heat it up and you melt it. You get it really, really hot. And then you pour it. And when you pour it, you're going to get this meshwork of polyacrylamide, right? Just looks like a meshwork. So what happens is you're going to load a sample onto the gel. Now, this sample is going to travel down the gel, but what's going to be dependent upon how it can travel is its size. So the bigger it is, the harder it is to get through the gaps, right? Because there's no pores. The smaller it is, the quicker it just slides right through. That makes sense? Okay, so there are two different kinds of page gels, and I don't like the way your book says it, so we're gonna do it a little bit different. So you can have a native page gel, or you can have an SDS page gel. Okay. In a native page gel, what happens is your protein of interest is going to run based on size, but the reason the proteins will run on the gel is because they have particular charges. And you have a positive charge at the top. Whoops. You have a positive charge at the top, and you have a negative charge at the bottom. And so proteins that are negative will migrate towards the negative, right? So a native page gel will run on charge as well, and conformation. If it's packed really tightly, it's going to travel more quickly. If it's more of a spread out protein, right, it's going to run more slowly. So native talks about the characteristics of the protein. We haven't messed with the protein very much. With an SDS page gel, here you're going to add SDS to it. And SDS is a substance that will coat your protein in negative charges. If you coat your protein in negative charges, what's going to happen? Do you think it's going to keep its shape? No, you're going to lose the shape, okay? So conformation is not going to be a dependent factor anymore. Will charge be a dependent factor anymore? No, because it's really all going to be negative. They're all going to be, all the proteins are negative. So all the proteins are negative. The bigger the protein, the more SDS coats it. So what is it going to run based on? What's left? Size. Just runs based on size. Size only. Okay. Now, you have to think about if you have a protein that is made up of more than one polypeptide. Usually those polypeptides are held together by covalent linkages. So SDS is not going to break those covalent linkages. In order to do that, you have to add a reducing agent, right? Because what kind of a linkage do we have to hold polypeptide chains together? What's the most common? Between two cysteines, a disulfide bond. So if you add a reducing agent like beta mercaptoethanol to your sample, you'll break those bonds as well. Okay. So some people, before they run an SDS page gel, they'll add beta mercaptoethanol and some don't. It just depends on if you want to break those disulfide bonds. So that's kind of the other thing. Okay. So let's look at a sample. So here's an example of a page gel. Doesn't tell you if it's native, doesn't tell you if it's SDS. Okay, so we're going to just look at how a page works first. So what you have are, and I thought the book corrected this, but it didn't. It's a positive charge at the top and it's a negative charge at the bottom. No, I take it back. I take it back. I take it back. It's negative at the top and positive at the bottom because all your samples are going to be negative. All are negative with SDS. They did fix it. Okay. So you put your samples in these little wells. And this, do you see the little solid light blue? The solid light blue is your polyacrylamide gel mix. So this is the spaghetti strands that are all twisted together, right? And your protein has to get through that spaghetti mixture in order to get to the bottom. So you're going to load them at the top and you're going to put some dye in with your protein sample. And you turn this on and you walk away. And typically you put a ladder here. So a standard set of proteins that you know are a particular size so that you can have a reference. And then you run your samples. Do any of these lanes have a purified protein in them? If each lane is a sample, do you have one protein in any of the lanes? No, right? They're all mixes. They're all mixes. So whoever did this purification should probably go back and replan. But basically, here's your sample, right? And you add dye to it. When you put it on the gel, if it's all negatively charged, it's going to run towards the positively charged anode, right? So you're going to separate based on size. Your larger proteins will be at the top, your smaller proteins will be at the bottom. So how do you make sure everything is negative? You use SDS. So sodium dodecyl sulfate is a detergent, right? What's a detergent? Like something you wash your hands with, right? How does that work? All detergents have a fatty acid tail and then a charged head group, right? And so what happens is a fatty acid tail is going to associate with all of your nonpolar portions of the molecule, right? And then the head groups are going to stick out and they're going to coat whatever protein it is. Doesn't matter how big, how small, whatever it is, SDS is going to coat the whole thing. So you're going to end up with an equal charge to mass ratio. The larger the mass, the more charged SDS particles you have. So what's going to happen when you run a current through your sample? The bigger things with more charge are going to run. They're going to run slower. The bigger things are going to run slower because they're going to have a hard time getting through that matrix, but they're all going to run in the same direction, right? Your little ones are all negative. They're going to run through, but they're going to go through faster because they are smaller, right? Okay, so let's look at an example. So here's an example, right? What we've done is next to the sample, we've written the molecular weight of the protein. So if you just put a little bit of polyacrylamide, a little bit of spaghetti, what's going to happen is that you're going to be able to separate things that are really large in size. So a low percentage, you can separate large molecules. So that's at a 5%. We have a big separation between 250 and 150 kilodaltons. Switch over to the 15% polyacrylamide. Where's the difference between 250 and 150? They're really close to one another, right? So if you kind of know ahead of time how big your protein is, then you should know what percent polyacrylamide, right? As you put more spaghetti, it's harder for the big proteins to get through. But what about the little ones? Oh, now I can get some good separation between my little proteins. And so you can look at the molecular mass of the proteins, and you can correlate that to a migration distance in the gel. And if you have a set of knowns where we know they're a particular molecular weight, we can plot those. And then if we look at the distance that our unknown protein has traveled, and we plot this on the curve, we can estimate what? The molecular weight of our unknown, right? This is just like a standard curve. But this standard curve, we're looking at distance that these proteins have migrated versus their mass. And then all you do is take your unknown and estimate your mass. And so you can tell if you got it or not. Okay, so here's an example, right? We've done the whole purification scheme and we want to check every step. Did we do it right? So we have an SDS page gel. So this is an SDS page gel. What we have here in the first lane are our known markers, right? So we know this is 202 kilodaltons. We know this band right here is 121. We know this one is 79. So I take my cells and I don't even open the cells up. I just run them on the gel. Well, most proteins are where? Are they inside the cell or outside the cell? Most proteins are inside the cell, right? So you're not going to see a whole lot of protein there. But here, the lysate, we've broken open the cells. Now, these cells are probably like those bacterial cells that I said can make a protein of interest, like insulin. So let's just say I had bacterial cells and I programmed those bacterial cells to make a ton of insulin. Well, here's all my proteins in the cell. This one would be insulin. But do I have other stuff? Absolutely. This one's probably actin. It's one of the biggest bands in the cell. And so what I want to do is then I want to start my purification process, right? And so in this one, it looks like all they did was put it on a column, which I don't agree with, but looks like that's what they did. They put it on a column and this was the flow through, right? So maybe they put it on a chromatography column. So this was the flow through. Then they washed the column with a little bit of buffer, and then they eluted from the column. And so when you added that elution, so maybe it was the competing binding partner, or it was the ion exchange column, and it was the same, an ion of the same charge. Whatever it was, you eluted it. Okay, well, it came out in the first elution. That's perfect. All your other junk came through on the flow through, and this is your protein of interest. This is your elution. So this is a good setup. I don't believe that those are the only steps they did in purification, but that's what it looks like. So this is just put next to it some kind of column chromatography. C-H-R-O-M-O-T. Oh, I cannot spell today. Column chromatography. Okay, so they did it. They did it. Yay! All right, y'all are good to go. Have a good day.
Dr. V,
All right. Anybody need anything on the chat?
I have a question.
Good. All right. Y'all have a good rest of your day. Wait, yes. Go ahead, Sam.
Can you hear me?
13th, Wednesday, there's a train for work. I could probably still Zoom. Is that okay?
If you want to watch the recording, I mean, like, I know y'all have to do that for work, do it for work. If you want to watch the recording, you can. If you want to be live so you can ask things, that's cool. Doesn't
I
matter.
just didn't want to count as an absentee.
Just when, because it's going to mark you as absent, send me an email after. When it pops up as I was absent, I'll go in and I'll fix it.
Okay.
And I'll make an excuse.
Okay. All right. Thank you.
But don't forget to send it to me after it happens.
All right.
But no problem. Actually, yes, tomorrow at 8 o'clock, correct, Sam?
You do not agree.
Good, how are you?
I had drill over the weekend, so I couldn't really check the homework. Can
Okay.
you open it up?
I'll send you an email. I'll read it.
All right, appreciate it.
No problem. 8:30, yes. All right, Sam, I will see you tomorrow morning. Anything else, Sam, before I close? I'm sorry. Okay, it must be, I think she left. Okay.